Good afternoon, everybody. Welcome to the Community for Global Health Equity Brown Bag series. This is unusual for us in that we are completely virtual, but these are new times, so it's good for us to try something different and adapt. So my name is Kasia Cordas. I co-direct the Community for Global Health Equity. And today it's my pleasure to welcome and introduce Dr. Anirban Dutta. He is an assistant professor in the Department of Biomedical Engineering here at UB. He directs the Neuroengineering and Informatics for Rehabilitation Laboratory. And in his lab, he's been conducting interdisciplinary research in neural engineering, which is the application of engineering to neurosciences and neurological or neurophysiological problems. His research is multidisciplinary and has the overarching goal of treating, curing, or even preventing neurological disorders using something that he's calling neurosciences, which are bioelectronics that stimulate the nervous system. His research has been directed towards better understanding of neurophysiological mechanisms associated with relearning of visuomotor function, as well as other applications. And that's what he will be telling us about today. So thank you, Anirban, for making the time to share your research, and whenever you're ready.
Thank you, Dr. Cordos, for such a nice introduction, and thanks to CGHE for funding me. So initially, this work started, as I will explain in my talk, how this kind of started and where we are now. So it kind of initially started with funding from CGHE and in my lab. So we have been doing this work primarily on the animal studies and then some clinical studies that's being done in Europe and in India. So let me actually stop the video because I think it may slow down the talk, and then, okay. So now, so this talk is on this portable neuroimaging of neurovascular coupling in brain injury in adults and neonates. So as I said, so this kind of work started with the funding from CGHE, and the concept was developed while I was in Europe, working on my master's at the Charité. So there we had this concept of evaluating neurovascular coupling. So, and the question was why what's the importance of the neurovascular coupling? So, so a lot of this brain dysfunction is essentially a combination of dysfunction in the neural activity and then also the blood flow response, and they need to be coupled together for the neurons to work well. Because so when we say brain activity or the neural activity, that needs energy for it to create the networks that we use for our function, and those network needs energy which is then supplied from the by the cerebral blood flow, that's oxygen and the metabolites. And this coupling between the neuron and the blood flow, that's the neurovascular coupling. And also there's this coupling between the neuron and the energy metabolism. Now, the oxygen and the metabolites that are supplied by the blood flow, they need to be broken down or need to be synthesized in a form that neurons can use for their activity. I mean, as an energy for their activity. That's ATP. And so that's done in mitochondria, and that's why kind of create an integrated system where this coordinated interaction between neurons, glia, and vascular cells are important for the brain to function properly. Now, so being an engineer, so my goal was more to kind of look at what engineering techniques are available to understand this mechanism, right? So this neurovascular coupling and metabolic coupling mechanism. So, for example, we already know from prior works that neural activity can be detected using electrophysiology techniques, for example, electroencephalogram, local field potentials, and spikes. The energy metabolism can be detected with the pH changes in the brain, the state of the cytochrome C oxidase, which is the state of the mitochondrial oxidation state, and also the blood flow, which can be measured, of course, with MRI kind of techniques, but also non-invasively at point of care using portable neuroimaging that I will kind of discuss in more detail, which measures this deoxyhemoglobin and the total hemoglobin content. So basically my talk is primarily geared towards the engineering techniques which are used to identify any dysfunction in this neurovascular coupling in health and disease. So that's the idea. So the basic hypothesis kind of started from my work with Dr. Dreyer. So Dr. Dreyer, he at Charité in Berlin, so they have been working on this for a long time, where they're trying to identify this what happens to this neurovascular coupling in severe brain injury. So so in brain injury, usually there is a dysfunction in neuron that we know, and we identify that, for example, we can identify that using electrophysiological technique, which is, for example, EEG or ECOG, which is a invasive measure of the neural activity, where there is this slow change in the membrane potential. So that's what I'm showing with my mouse here in the bottom panel. So you, in the middle, you see this kind of like a dip, and this is actually very slow changes that happens in the electrophysiological signal. And then because this is an indicator of the energy need of the cell, so in healthy tissue, there is an increase in the blood flow, and that provides the energy that is necessary to bring this to the normal state. So this transient state, dysfunctional state is recovered, and then it's back to the normal state. However, in a very severe case, this is another extreme, which is where you have this depression, that's the slow potential changes, and then that, however, leads to an inverse hemodynamic response, where you have, instead of a hyperemia, which is increase in blood flow, you have a ischemia, or a decrease in blood flow, and that indeed is not what we want now, because there's a need for energy. And because of that, this slow potential changes, they last longer and eventually lead to injury to the brain. Now, so on the left is this healthy tissue, where you see this blood vessel, the arteries supplying the blood, the capillaries here, and then that's kind of in network with the neuron through this astrocyte. So that's kind of the network here. However, on the right, what you see is that in this inverse hemodynamic response, you have this vessel has constricted, and then the neurons have swollen because they didn't get the energy to maintain the ionic balance. Because of that, the neurons have swollen, and this is a state where there is a tissue at risk of damage or death. So this is basically the idea behind the development of the engineering techniques that we then use to identify this state, or a transition from this healthy tissue state to the tissue at risk state. Now, this is in general, so there's a lot of work, recent work in this area, and then one can look into this review by Adekola, which kind of says that this neurovascular dysfunction can kind of take a lot of different shape. For example, there is a reduced cerebral blood flow, like I said here, which leads to a hypoxia, which can be a transient hypoxia, causing damage to the tissue. Then it can also lead to this blood and brain barrier dysfunction, which can lead to an altered homeostasis in the brain tissue. Trophic failures lead to the cell vulnerability to death. And of course, recently there's this idea of the reduced clearance of the byproducts, especially this protein accumulation in the brain that can lead to dementia and all. So this is overall kind of like a like scientifically an area of interest to different for different applications, and for different researchers interested in different issues, health issues. What we are in our lab is interested in is to develop this technique, which is, I will go into more detail, which is basically a portable neuroimaging technique, which can detect the state of the vessel. So how much the blood volume and then the state of the neuron, how the neurons are firing, and then to kind of identify using signal processing techniques, the coupling between the two. As you can imagine, this coupling would be transient, so there's not a stationary signal. This basically is transient signal that we have to kind of accommodate with our signal processing technique. And any impairment in that coupling can lead to, for example, in brain injury and unresponsive patients in coma. And we hypothesize, or our postulate is that this recovery of this neurovascular coupling will precede the recovery of consciousness. So this kind of would be a marker of good prognosis. So this is the basic hypothesis that my lab is working on to develop these techniques. Now, the first application that I wanted to mention is this an application in neonatal hypoxic ischemic encephalopathy. So this is so after I left Europe and I came back to UB. So I was funded by this Committee for Global Health Equity to develop this portable brain scanner for the detection and management of neonatal hypoxic ischemic encephalopathy, so HIE. So this was so engineering work and computational work was done in USA at UB, and then the clinical study was run in India by Dr. Vijay Das in a low resource setting. So at UB, so Radhika initially worked on this as an intern, CGHE project intern, and developed this this sensor and did a lot of bench testing to test different LEDs or different parts for this sensor and to identify the best combination. So that's how it started. And then Brian Hagen, who was a master's student, did a thesis on this, primarily working on the signal processing pipeline. However, at that time, we didn't have the data from India. We were setting up the study in India. So we used preclinical data from in collaboration with Dr. Nair and her team at UB. So this was the data from an animal study, and we used that to develop the signal processing pipeline. Then eventually the clinical study got registered in India, and we then used this signal processing pipeline and the hardware that we developed at UB to collect data in India. So this is what we call the Cerebros, which is now also being developed as a product by and then the signal processing and the biomarker identification using the data that we collect in India has been done by Sandeepan Bhattacharya at Argomedtech. So this is the state of this this project. So now to kind of to go into the details, like what are the features and all, I would like to discuss a little bit about this this hypoxic environment. So this is based on this prior work. So there are a lot of work in this area that has investigated the state of EEG in hypoxia, so in lack of oxygen in a brain tissue. So this paper specifically, especially by Goel, kind of identified this spectral dispersion, that is this the change in the frequency content in or the power in this 3 frequency bands across this different states. So at baseline, then you have the hypoxia, and then after that the asphyxia, and then after that the recovery. So how this power distribution changes across this 3 frequency bands. So what they noticed is that so during hypoxia, the relative power of the medium and high frequency components, that is that 9 to 14 hertz and 18 to 21 hertz, that increased by like more than 150%, 160 and 176%. And then, so that's in the hypoxia state. And then in asphyxia state, so that's when this medium and high frequency powers that increase even further to 280 and 400%. So this distribution of power across frequency seems to be a feature that we can use to identify this transition from hypoxia to asphyxia. But after this initial increase in power, then eventually, like within 40 to 80 seconds of asphyxia, the total power of the EEG signal decreases substantially, drastically, and it almost flattens to 0. So that you can see here on the picture on the right. So this is what we are interested in capturing using signal processing techniques. And of course, since we had near infrared spectroscopy, so we could see what's happening on the blood flow or the hemodynamic side. So that's kind of was the goal of Brian Hagen when he started his master's thesis. So um, so now, just briefly, I would like to explain this Lam model. So this has been explained very well in this video, in this Jove publication by our collaborator, Dr. Nair, and her team. So what we are looking at. So this is a study that's they have developed for this newborn resuscitation. However, we used only 3 signals from here. So this model is this this model kind of implements this hypoxia, then asphyxia state, and then recovery from that using chest compression, where we can see this transition back to this spontaneous circulation. So this has this whole transition that we can look into using film processing techniques. And the signals that we were interested in is the EEG, which was a differential EEG from a single channel differential EEG. Then we have NIR signal, near spectroscopy signal, and then we have this carotid artery flow. So that also we had the signal. So based on this 3 signal, we try to see if we can classify those different states that the neonatal Lam model kind of goes through during the experimental procedure. So here you can see, so there's a cord clamping leading to hypoxia, and then there's asystole when the heart activity, when the subject loses heart activity, and then there's chest compression to revive or resuscitate the subject back to spontaneous circulation, and then you have this state that we then wanted to see if it's really if the subject has really returned back to this initial baseline state or not before cord clamping. So that's kind of the idea behind it, and you can see here. So as expected, so you have some baseline measure, and during asystole, the blood pressure, the carotid flow, they all come to close to 0, and then we couldn't use the data during chest compression because during that time there's a lot of artifacts, movement artifacts, so we had to remove that data. And then what we looked at is this the data after return of spontaneous circulation, ROSC, and how the EEG and blood flow is doing at that state after this perturbation, asystole, and the chest compression perturbation to the subject. So that was the idea, and this is kind of the sensor that we repurposed for this experiment. So this is based on our hardware, but the sensor was repurposed for this particular animal study. Now, Brian looked into this 5 subjects. So these are basically of the gestation, like 140 days, 140 days gestation, and then there are so there's 1 female and 4 male, and so basically this is the data that we received from Dr. Nair and her team while they were doing their own studies. However, we were interested in investigating this EEG, especially the seizure activity and all in the animal model in the subject, and how that kind of recovered after the perturbation. So the phases that this experiment had is this baseline, which is the control. So that's the initial state, and then there's this cord clamping leading to a hypoxic environment, and then the heart stops. That there's asystole, which is the no heart activity. Then from there to resuscitation. So this resuscitation phase was not used because of the movement artifact, and then after that, the subject return to back to the spontaneous circulation. That's 10 minutes of data that we use for this analysis. And again, there's this 3 measurements. So this differential EEG measurement, carotid artery flow, and then the oxygen saturation. Okay, so I won't go into too much detail on the data processing workflow. However, just to kind of highlight the main points. So we had this raw data, and then of course, because of the because of the artifacts and the noise in this in the laboratory setting. We had to do a lot of pre-processing to clean the data. Then after that, the first was, so Brian first worked on this classifier. It's an LDA, a linear classifier, that was for identification of ictal activity. So that was the classifier that was kind of first tested on this an open source database. It's a human open source database by MIT, and then which has already been labeled by physicians, so he could use that to test his classifier, and then we use that classifier to to on the animal data, and then so we did what is called non-regressive modeling. So we kind of captured that power spectrum using, we already parameterized that power spectrum using a model. That's a AR modeling. It's kind of a technique that's used in signal processing. Then we use that for classification. So the first is basically to see if this LDA classifier can pick this ictal or pre-ictal activity. So in the time series, so these are basically kind of like, seizure-like activity. Then we remove that, and then after that, the data that's left is then goes through an SVM, support vector machine classifier. And the goal was to identify this hypoxic state. And this is basically what we wanted for our study in India, because in India, like maybe, so in this animal study, we have this extreme case where we see a lot of seizure activity. However, in a mild to moderate cases, we were interested in kind of identifying this hypoxic state, and if we can kind of classify that, the severity of that using this classifier. So that was the goal, and then this was then used for validation. So that's the data processing pipeline. So I want to give you this one example here. So this black on the top, so this black, this signal is the EEG activity. Okay, so this is basically the baseline, and then as you can see, during initial handling of the animal or of the subject, you have this this change in the flow. So this green is the carotid flow, and the red is the oxygen influx based on the oxygen saturation. So and then you see there's a lot of this transient changes, and also there's some the seizure activity even before the actual perturbation, which is here after 4000. So this is where the asystole. So even before that, there's this seizure activity, and then we have this asystole, and then the baby or the subject then recovered after that. So we removed the data when this chest compression and the resuscitation was performed. So now the first goal was, so this whole data set that we have now, the first goal was to kind of identify if we can identify the ictal activity. So that was done using this classifier that Brian developed. And we first removed all ictal activity from the data. So all this, so the yellow you see here, they are basically all ictal activities. They were all removed. And after that, we are left with the control, or the control is in blue, so that's the baseline, and the green is green is the hypoxia. So we now have to separate this hypoxia from control, and that was done using an SVM class support vector machine classifier using again the AR parameters or autoregressive parameters. So this is basically the idea behind this approach. It's a two-stage classification. Now, just briefly, so these details are there in Brian's thesis that's online. But so we did very well in detecting this ictal activity or seizure-like activity, where the accuracy was around 92%. However, this hypoxia activity using just EEG, we could detect only with an accuracy of 78%. So this is only with EEG. So we didn't use the hemodynamic signal here. So that's the first. So that's the development of the signal processing, and then we applied this to the data that we collected in India. So in India, this study was funded by Gates Foundation, and we had this neonates with gestation greater than 35 weeks, and then greater than 1800 gram. So this was done in an ICU setting. We are now working on a community setting. It was done in an NICU setting where these neonates were admitted for the management of perinatal asphyxia. And also we had the controls who are the babies matched for gestation and weight. at the NICU. So again, exclusion criteria was premature babies, less than 35 weeks, babies with multiple congenital abnormalities. Then if the parents didn't give consent for the study, then also those babies were not included. And then the total study participants were 60, so 30 in this parental asphyxia group and 30 in the healthy control group. And the duration provided by the Gates Foundation was 6 months. So this is a study that we did in India. And so here, this is basically a clinical study where the standard technique used for scoring hypoxic ischemic encephalopathy with HIE is the Sarnath scoring. It kind of divides it into normal or mild and moderate and severe HIE. So it looks at different aspects like level of consciousness, spontaneous activity, posture, tone, primitive reflexes, autonomic system like heart rate, respiration, people. So this is kind of a standard technique used by clinicians to classify, and we use that to kind of stratify the babies in these three different groups, and then wanted to see how our neurovascular coupling features are clustered across those three different groups. So this is the hardware that we used. So it's a low-cost hardware that we developed for India. This has the simultaneous. You can simultaneously measure near spectroscopy signal, which is using this two LEDs, two photodiodes, and then we have this electrodes for EEG. So this is kind of, so this is this basically, as you can see in the video here. This is time multiplex. So this LEDs, they blink alternately, and then we collect the light that then passes through the brain tissue, detected or measured at the photodiode, and then based on the signal that we get there, we can do some signal processing to identify the oxy and deoxyhemoglobin content in the brain tissue. And also we can measure EEG. We also had, so this is basically so it's basically all integrated in a headband. So this is a wireless unit with the sensors integrated in a headband, and that can be tied on the head of the baby in an ICU setting, in an ICU setting. And since it's wireless, so we don't have to be in that room. We can, I mean, here the laptop is in the room, but we can also collect from outside of the NICU. So we here in the study, we also added some extra EEG electrodes using one of our other devices here. So it's basically the EEG, so primarily from this central parietal area, and then with overlapping FNIRs, which is 2-channel FNIRs signal from that area. So this is the setup that we have for India. And then the preliminary results, what we saw is that so we applied this ARX modeling that was developed at UB. And we were particularly interested in this EEG bursts. So in general, the signals, so the human data was not that severe as an animal data. So we didn't see like a lot of seizures and all, although we did see some seizure-like activity. And then we were interested in what's the blood flow is doing, or what's the hemodynamic is doing during those EEG bursts. So that was kind of the goal of this analysis. So what we found is that in few severe cases that we had, there was this lot of this negative neurovascular coupling. So when there's an increase in the energy in the EEG signal, there is a decrease in the total hemoglobin in that tissue, in the nearby tissue. So that's if it's more than 80% in the severe HI case, and in severe HI case that led to frequent this burst suppression, and unfortunately the prognosis was really bad in these babies, few babies that we had. And then in the moderate HI, this negative neurovascular coupling was between 50 to 80% during the EEG burst. So we only looked at first we did use Brian's classifier to identify the EEG burst, and then look at what the hemodynamics is doing during the EEG burst. So that's what I'm showing here. So in this moderate HIE, there was less frequent EEG burst suppression. There were frequent seizures, when compared to the severe HI. But this neuromuscular coupling, the negative neuromuscular coupling was less frequent in moderate HI, and it was less than 30% in mild HI during EEG burst. So this is something that we are now kind of proposing as a as a as a marker of the severity of HI. Now, so Yashika, now I want to kind of highlight this work by Yashika, who visited me at UB, and then who is working on this computational modeling to look into what this kind of negative neurovascular coupling does to the tissue. So, for example, there's a lot of this clinical evidence that this brain tissue after brain injury is susceptible to epilepsy, early epilepsy. Then the seizure activity leads to an increase in extracellular potassium concentration, also glutamate concentration, and how that affects the, in general, the generation of seizures and the seizure frequency. So that's, but this is a modeling study that we have started with Ishika, who is a PhD student in our collaborating lab at IIT Mandi, led by Dr. Subhit Roy Chaudhary. This is one of the work that's going on with this data that we have collected from animal as well as from human. Now, I wanted to kind of now highlight the second application, which is in Denmark. This is a disorder of consciousness study. So it's an adult application of this system or the signal processing. So here, I won't go into too much detail of this study, but the basic idea is to kind of identify using near-CEG the state of consciousness and the prognosis, and if we can identify some prognostic biomarker of recovery. So these are brain injury patients, primarily cardiac arrest patients who are non-responsive. And this is a clinical challenge because a lot of these patients get misclassified as unresponsive, being unresponsive and in a vegetative state. So we wanted to see if we can use this technique. Now, like I said, so the motivation is primarily to detect this consciousness or covert consciousness in this patients in a neuro ICU setting. This is important because a recent meta-analysis showed that 15% of these patients who are considered to be in vegetative state can follow commands. And this was done with fMRI. And then we are applying this, our fNIRS-EEG technique to identify to see if we can identify these patients with this covert consciousness who are considered to be in vegetative state, but may not be. So the primary objective of our collaboration is to use this fNIRS functional NIRS EEG technique or signal processing in a serial multimodal evaluation while the patient is in ICU in a non-responsive state and is subjected to different active paradigm where the person is being asked to do some mental imagery task as well as passive movement and also resting state. So we are kind of doing a serial multimodal evaluation to identify if we can find any biomarker of covert consciousness as well as prognostic biomarker for the recovery of these patients from unresponsive state. The secondary objective is more scientific to kind of see if this neurovascular coupling is kind of a marker of recovery of consciousness after acute brain injury. So this is the two main goals. Now we did it. So the preliminary study that we already did, and this is actually is under review, is this resting state fNIRS EEG assessments. This was done in a convenience sample at a neuro ICU and at Rick's Hospital in Denmark. We had 9 who were unresponsive patients. And we did this fNIRS EEG assessments on them. Again, the exclusion criteria was any kind of injury or life-threatening, any kind of scalp injury or life-threatening cardiovascular instability. We also had this conscious patients who were also in a step-down unit, in a hospital setting, but in a step-down unit, but they were conscious, and then they served as control. So again, the level of consciousness was estimated here by board-certified neurologist based on this 4-scale full outline of unresponsiveness, which categorized them in like the states, which is coma, then the vegetative state, minimally conscious state, minus minimally conscious state plus, and emerged from minimally conscious state. So this classification is based on like visual and motor subscales. So so it kind of is a fine-grained classification of consciousness in these patients. And the data was stored in a dedicated database there, and then the computational analysis was done there. Okay, so now quickly the setup that we had is we had this EEG in the frontal, prefrontal, frontal, central, and parietal areas. We had NIRs which were co-located, and then we did this data collection in 15-minute segment for 45 minutes. Because the nurses, they had access to this patient, and so we had to have this small segments of data, but we did it multiple times to get about an hour of data for each subject. Now so now kind of briefly, we have this model that we are developing. So here, one of the challenges is that the data is transient. So the IC analysis that usually is done is kind of fits one statistical model to the whole data, which is not applicable here because the person can go out in and out of different states of consciousness. So we use this adaptive mixture independent component analysis AMICA model, where you can use this, so where it actually fits different models. So it divides the data, segments it into different regions of local stationarity, and then fits models for each, which can be different for different time segments. And the idea is to kind of look into the probability of those models. And some of these models are, of course, artifact and that we need to reject. But then some of them are physiological, and then we can use them to classify the state of the brain. So here it's kind of shown here, we have 3 models, and then they kind of, so there's a time series data, and it goes through different states in the time series data. So again, this is the kind of like the kind of an overview. So we have the EEG data, we use that using AMICA, but you also have the NIRS data, and then we use this neurovascular coupling approach to identify biomarkers that we can then combine with the EEG biomarkers to identify the state of the person or the conscious state of the person. So there are a few things that we noticed in this small data set that we collected. So the EEG power in this 11, 22, and 34 hertz, the alpha and beta band activity was primarily centered at the right frontal areas, whereas the low frequency 2 hertz and 6 hertz activity, delta and theta activity, was on the left frontal areas. And so the important thing that what we found is that using this multimodal model AMICA, we had 5 models. So the 2 models were artifacts that we rejected. We are left with 3 models. So here you can see that so different colors represent different models. So this is one time series from a subject, a patient, and then you can see that the person kind of goes through different states while in this unresponsive state, or a state transitioning from unresponsive to minimally conscious state. So the person kind of goes through different models, and those models can classify. So here, for example, on the right, I'm plotting on x-axis is the probability of model 1 and y-axis probability of model 2, and that goes from 0 to 1. So this model 1 and 2, these 2 models did very good in identifying, or these 2 models were active in this class false, which is this conscious controls. And conscious controls, model 1 and 2 are active, and you can see they kind of together share the probability. Now, if you then look into this model 2 and 3, then they are primarily active in this low or unresponsive patients, a 4 score less than 7, and they kind of share the probability together. And then so model 2 is kind of like the transition between the two. So model 1 is primarily red here. So that's the conscious control, and then the green is our unresponsive, low responsive patients who are primarily model 3. And there's some overlap, so there's like some green here also, but it's primarily separated. So this actually kind of helped us significantly to separate this different conscious levels in the patients. So the other thing that I actually wanted to also show is this this initial neurovascular coupling. So the patients who had neurovascular coupling was good initially. That's basically based on this wavelet coherence between 0.07 hertz and 0.13 hertz in frontal area. So who were high initially, they all recovered consciousness. However, unfortunately, the patients who had poor neurovascular coupling at the admission, they had a poor prognosis and they didn't recover consciousness. So, so this is one of the things that we are now looking into with a larger data set, and we are collecting data. But this is something that we think is important for for this neurovascular coupling application in prognosis of recovery from unresponsive state in acute brain injury. Now, so this is basically the data that we have, and we have all of this is now under review. Now, some of the work that's ongoing and which is currently at UB with Dr. Tomita, with Dr. Leddy and Dr. Sisto, and also in UK at NHS and in India at AIMS and IIT Gandhinagar. So what we have here is this system and signal processing that we have developed, and we are trying to see if we can apply this in stroke, concussion, mild cognitive impairment, etc. So I will just quickly give you some preliminary hints here or the results. This is again unpublished. We are still collecting data. One of them is this, what is called a neurovascular stress test. So here it's similar to a cardiac stress test. So where a person walks on a treadmill and slowly the difficulty is increased and the heart rate increases, and then it's allowed to reach to the maximum heart rate, which is this AHA criterion, which is roughly 220 minus the age. And some patients or some subjects can continue exercising until that maximum heart rate, and their neurovascular coupling doesn't deteriorate, and they reach the maximum heart rate. But in some, there is an early deterioration of neurovascular coupling quite early before reaching the maximum heart rate, and then they cannot even go to the maximum heart rate, and the exercise has to be stopped. And this is what we are considering as like a marker of dysfunction. So this early, so here on the right, if you see here, so the red is this neurovascular coupling. It's kind of improves initially with exercise, but then it then decreases. And this deoxyhemoglobin continues to increase in the brain tissue. And at some point they cannot continue the exercise anymore, and they asked us to stop the treadmill. So this is this neurovascular coupling stress test for physical exercise. Then we have this cognitive exercise test, where they are given a cognitive task, which is this LASSI task. Essentially what this task is that they are given a list of objects to remember, and after a brief delay, they are then asked to recall those objects. And this list can be short or long to increase the difficulty level or to adjust the difficulty level of this task. Now, what we have, of course, with the longer list and with more difficulty, there might be what's called a semantic intrusion. So they actually add objects which are not there in the list. But what we are looking at is basically this neurovascular coupling, primarily in this dorsolateral prefrontal cortex. So this area gets active during this kind of a task, which is basically the encoding task when they try to remember this list, and then recalling task when they try to remember. Again, we are looking at this neurovascular coupling deterioration with an increasing difficulty of the task. So this is again kind of like a stress test, but for a cognitive task. And then the third thing that we are recently looking at is this COVID-19. So COVID-19, so we all know that this is basically respiratory distress syndrome. So this is basically primarily to do with the lungs and the respiration. However, recently there is this some evidence of it affecting the brain. So this is basically a recent work by this Filatov. It's a case study, and we have now started looking into this using our device and the signal processing method. So we already know that there is something called sepsis-associated encephalopathy, SAE, which is a transient reversible brain and can have this brainstem effect in patients. And especially in COVID, there is sepsis syndrome. So we are trying to see if that can be identified early using brain imaging. Especially since if the brainstem gets affected, then that actually can lead to respiratory distress. So it may be that the brainstem is affecting the respiration. So anyway, so this is something that's very new, and we have just started applying this for this COVID-19 cases. And this is important because in SAE, we already know very well that there is this possibility of neurovascular uncoupling, which is due to this microcirculatory dysfunction and low blood flow. And this can, of course, be detected with fNIRS and EEG, and that's our goal. Okay, but last but not the least, I want to kind of quickly highlight this neurometabolic coupling. This is another application, but in a cerebral organoid cell culture study. So the same idea that can be used on human application can be used in cell culture application. This is a work by Stokowiak in this cell culture, 3D cell culture, where we are trying to find out the state of the mitochondria. So here there is no blood flow, but we can look into this mitochondrial state, which is the energy state of the tissue, and see what's the coupling of that with the neural activity. That's the firing that we are trying to, or neural firing, which we are trying to identify with this excitation inhibition balance. This is in schizophrenia, and some preliminary study that, or preliminary data that we have collected shows that indeed there is a relation. So in schizophrenia, there we found reduced activity of this cytochrome C oxidase compared to healthy organoids, and that was related to an increase in the excitation inhibition balance in schizophrenia, which is, so this again, we are working on a publication on this, but this unpublished data, and this you can see here. So control and schizophrenia, the slope is different. It's flatter here. It's minus 0.5 compared to minus 1.4. So it gets flatter, which is due to the increase. This is known to be due to the increase of excitation inhibition balance in the tissue. So anyway, so there's a different application that we are looking into with this hardware and the signal processing technique that we have developed. So the end goal is to establish this neurovascular and neurometabolic coupling as a marker of the brain tissue state, and if that can be used as a marker of recovery of consciousness in acute brain injury, in other brain disorders. So we have, of course, now a very low cost, low density hardware that we are using, but we have to also establish if this is equal or if it's inferior to more expensive hardware being used in this like 28 channel or 256 channel systems. And so this is our kind of like the goal for all our ongoing studies. And we have now we are now looking into this stress test, which is under stress, how neurovascular coupling changes under physical and cognitive stress, and to see if that can somehow provide us some way to identify a mild cognitive impairment or other like vascular dementia or other brain disorders early on that are affecting the humankind. So this is like very recent work, and so yeah. So overall, so my lab is primarily working on this hardware and the software tools for different applications with clinician collaborators at UB, but also in Europe, in Denmark, and in UK, and also in India. So that ends my talk. Thank you very much for this opportunity. I can now take questions if there are.
Hi, Anirban. This is Kasha. Thanks for this great talk. It's amazing to see how much you've done, and you know, we've mostly talked about your work in the neonates, but you've certainly taken this to different applications. So thanks for sharing that. But I actually do want to come back to the hypoxia in neonates, yeah. And just ask you to talk a little bit in more detail about that work. So you had presented that you had done the work in ICU in India and had found that EEG had 70% accuracy of detecting hypoxia. And I wonder, you know, you also used Effner's in that study, I think, as well. So can you talk about where that has gone, and you know, in terms of the relative specificity and sensitivity of that system in identifying infants, especially in very low income settings, what the applicability of that is. So so is that something that you can describe in more detail?
Sure. So so yeah, so we initially, so this is this classified as performance is shown based on the animal data, so the preclinical animal data that we have at UB, and then after that, so in human babies we cannot do an invasive recording or invasive measure of hypoxia. So that part is actually something that we cannot classify based on any gold standard. So what we did is, instead of that, we used the clinical clinical technique. So which is just Sarnat scoring, which doesn't really tells us much about what's the state of the hypoxia, but more this hypoxic ischemic encephalopathy and the symptoms of that. So it has this different level of consciousness, and also we kind of from animal study where our goal was to detect this hypoxia, which was clearly classified based on the invasive recordings. We now have moved to this classification of this Sarnat stages, and there what we have found is that uh, so this data is still under process. So initially, so the animal study was done in US, but then the human study and the data processing is done at the center in India, because they have funding from Gates Foundation, and we cannot use that funding, so we have to hire in India and then to kind of get this done there. So we have provided them with the signal processing pipeline and everything, and the I think we have now about 25 babies already processed, the data already being processed. And what they have seen is that this negative neurovascular coupling in the severe HI, so it's greater than 80%. So this is kind of like you can consider this as a biomarker. So if we see the 80%, the neurovascular coupling negative greater than 80%, there's a chance of severe HI. Now, what's the chance? That's basically the probability is with a small data set. We haven't computed the probability because it's not really a robust measure yet with the data set that we have, because we only had in the severe HI, I think 3 or 4 babies. So, but then most babies were mild. So we are very confident. We can say with the probability of more than 90%, more than 90% or 0.9, I can say that less than 30% negative neurovascular coupling will definitely mean that it's a mild HI, and the subject will recover. But then the moderate also we didn't. So the other 25, I think we have like about 5 or 6 or 7 moderate HI. So that's also this again between 50 and 80% of negative neurovascular coupling is a good indicator. But again, we haven't really done a very robust analysis on this probability. So we have to have more data, especially on the severe HI, which we didn't see that common. And also, I mean, all severe HI cases, unfortunately, the babies died. So we don't actually. Yeah. So this is currently the state, but they actually continue to, I think by the summer, there's a plan to submit this for publication. So because the Gates Foundation, that project is going to end. So yeah, I think that's the state right now. But whatever I presented, the data is actually based on this animal model that Brian used. And the goal that you asked, like what's the final goal, is to have this device. So this is a device that we are now, this device is now going through this regulatory approvals in India. So from April on, Indian government has asked all medical device manufacturers to register the devices for clinical studies. So we didn't do that for the initial part of the study, but then now we have to register it, and we are doing that currently to release it in the market. But I think we need more data to kind of come up with any kind of or to kind of establish this as a biomarker. So what we currently are doing is just providing the clinicians with the data so that they can look at our quantitative measures, but also can look into the EEG, for example, and their own clinical expertise to make the judgment.
Thank you. That's great. I don't know if there's, I have more questions, but I want to make sure that if there's other questions that we're able to address them in the time that's remaining. We have a little, like 4 minutes or so to conclude.
I think you should go for it, Kasia.
Thanks, Lisa. So you had said that there is a plan to continue to gather more data, and I'm just wondering. I mean, I think that this is so important to develop these devices for low-income setting, and but I'm wondering if there are specific diagnoses with respect to hypoxia in for example, in the United States, in hospitals, could you kind of take an approach from the other side and try to validate the device against something that exists here, and then be able to provide better probabilities given your readings and scoring for the lower income settings?
Yeah, I mean, so that's a good question. So for neonates, actually, it's very challenging to do the study in US. So that I have, so I've contacted a few clinicians. So in neonates, so in India, of course, we could do the study. In Europe also, it's there are groups which are doing it. And of course, this device is something that that we actually, it's basically wasn't manufactured from scratch. So these components inside it are CE approved. So, so this is basically based on OEM part, and then we kind of develop the sense, in the LEDs and the signal processing and put together the system. But overall, the device can be used. So in Europe, we actually are looking into getting it. So because the components CE approved, we have to get the approval for the sensors and all. But the clinical study as such currently for neonates, at least, we don't have any plan outside of India, just because of we didn't get any clinicians who can kind of allow this or can run this. Now, so we have now, that's why moved to adult, because this cardiac arrest in adult is an HIE, and in adult we can do this. So we already have this approved at RIGS in Denmark. So, and the signal processing remains the same. So we are using the same pipeline across these two studies. So the idea is, if we can establish this in adults in Europe, based on the data that we can collect at Denmark, and then to use that to identify, if possible, some biomarker. In India, we primarily are going more on this big data kind of an approach. So we are trying to see if we can, I mean, in India, we can collect, of course, a lot of data. So the idea is to see if we can identify some biomarker from that large data set. But currently, it has kind of reached a point where there's this neonatal work in India, driven primarily by this Gates Foundation, and we are trying to apply for this next phase. And then the adult study in Europe, which of course now we are planning to fund it. So we are kind of moving from neonatal to adult with this approach. So that's kind of where we stand now, currently.
Okay, thank you. One more chance for another question, otherwise we will need to wrap up.
Yeah, I'm good. Yeah, please go ahead. Yeah.
Okay, so I will take that as people are choosing to contact you via email if they have further questions. Thank you so much for your time and for presenting this impressive body of work. All the best with everything that you have going on and planned. That's quite a lot. And thank you for joining us on. Everybody joining us on Zoom, and have a very good day. We're gonna sign off now.
Yeah, thank you, Dr. Kodas, and thanks for this opportunity. I really appreciate it, and also the funding that CGHE provided to get this started in units. Thank you very much.
Thank you.